Okay, good morning to everybody in the US and good evening to all of you linking in from India. So, welcome to the students, parents, and teachers. On behalf of the International Visa Foundation, it is really my great pleasure and honor to be introducing our speaker today, Dr. Laks Iyer. So I'm sure you all have had a chance to look at his CV, but very briefly, Dr. Iyer did his bachelor's from Mumbai, India, in microbiology, and then he did a master's in biotechnology from Madurai Kamraj University, after which he got a PhD from Texas. He worked on plant virus interactions, and soon after that, he joined the National Institute of Health, which is one of the largest medical research institutes in the world. And since then, Dr. Iyer has made many great contributions to the field of understanding how proteins function and predicting protein structure functions and so on. You guys are in for a treat today because he's speaking on a topic not just that he's very informed about, but he's also very passionate about. So the one thing to know about Dr. Iyer is anything he does with great passion. He puts himself completely into it. So if you haven't had a chance to visit the little in his CV, there's a little website about his fold scope. And the stuff he does with that. He has a little home microbiology lab. I urge you to take the time to visit that. There is some amazing stuff and images over there. Dr. Iyer, with that, I won't take much time. Take it away.
We're going to have a very short program by the student, okay?
Yeah. Okay.
And then we will have the program by 1 second. I'm going to start sharing my screen and get this. Let me see. Ardra, can you stop sharing your screen? That might probably make it easier for me to, let me see. Can you all see my screen?
We see your screen. I'm not sharing anything.
Oh, okay, Ma. Good. That can you see the screen with the young boy there?
No, it's blank right now. All I see is like a black.
Oh, okay. Let me see in that case how I'm trying to figure out how I should share the screen. It says share screen.
And then you can pick what you want to share.
Yeah, I picked that and it says, okay, I said share. And let me see if that brings up, are you seeing now?
I think you're on the screen with the Zoom recording of this meeting because I see myself, like on your screen.
Oh, I don't know why this is not sharing this video. 1 second. Let me, in that case, again, I'm technically very poor. Oh, give me a second. Let me see if I can bring this.
Hi, I'm Akshay. In second.
Now I'm going to see if I can share the screen. Now,
Yes, now we see you. Now we see the
Yeah,
kid.
now it's visible.
Wonderful. Okay, give me a few seconds. Okay.
Hi, I'm Akshay in second grade in PSBB, Chennai. When we think of students, which great Indian comes to our mind? The hero of World Students' Day, Dr. APJ Abdul Kalam. In order to pay respect and remember him fondly on his death anniversary, that is on 27 July, I'm going to take an oath on his 10 commandments for students and youth. Let me start. I realize that a small aim is a crime. I will have a great goal in my life and work hard to achieve that goal. I will acquire the knowledge continuously. I will sweat. I will persevere to achieve the goal. I will work with integrity and succeed with integrity. I will be a good member of my family, a good member of the society, a good member of the state, a good member of the nation, and a good member of the world. I will always try to save or better someone's life without any discrimination of caste, creed, language, religion, or state. Wherever I am, a thought will always come to my mind. That is, what can I do for you? I will never get addicted to liquor, smoking, and gambling. I will always try to retrieve people from addiction and help them to lead a quality of life. I will always remember the importance of time. My motto will be, let not my winged days be spent in vain. Now I will plant at least 5 trees in my neighborhood and grow. I will always work for making my village clean, city clean, and state clean so that my earth will be clean and green. I will do all my best to achieve energy independence by 2030. As a youth of my nation, I will work with courage in all my tasks and enjoy the success of others. I am as young as my faith and as old as my doubt. Hence, I will light up then the lamp of faith in my heart. The national flag flies in my heart, and I will bring glory to my state and the nation. I will do all my best to follow these words. Why don't you also? This is Akshat signing off. Thank you.
Now, how was that?
That was
Really
lovely
wonderful.
and very
That's
sweet.
amazing, really amazing. Oh my God.
Lakshminarayan, the floor is yours, my friend.
Okay. Let me first make sure that my
Yeah,
screen is shareable, and then
please. Yeah.
uh,
Yes. Yes, we can see your screen. Yes.
So thank you so much, Dr. MS, and thank you so much, Ardra. I know you well enough to not call you Dr. Kachru, uh, for this invitation. I'm delighted actually to speak in this forum. More than anything, I'm even more delighted that this is a forum in honor of our people's president, Dr. Kalam, who singularly inspired generations of Indians across society to pursue knowledge and excel, as we saw in that video. And his boundless energy, even till his last breath, is very famous, and it is a great inspiration for Indians, people of Indian origin, and actually for the rest of the world. And it's an example for how we need to lead our lives. So it gives me a double delight to be part of this group. Now, I'm a computational biologist, actually, although my early training was in microbiology. So I shall not focus on my work today. It's a rather whimsical presentation of what interested me about microbial life. I don't profess this to be any kind of historical document or anything that is going to be super illuminating that you'll have to take down notes for. But however, I shall provide a perspective of what inspires me to study biology in general and microbiology in particular. And I have another question that's always fascinated me from when I was a child. I think there's some background. And
Kindly mute your,
Yeah.
please mute your phones, please. Please do not interfere with the lecture. Thank you.
Yeah.
Thank you very much.
So the other aspect that has obsessed me from when I was a child is what makes a scientist. Even though I'm a so-called professional scientist, I'm always curious to see what were the key reasons that somebody became a scientist, or how do we classify scientists. So I shall present some of that today in this presentation. So if this was a hall, this was very easy to do, but I'm going to now show you a set of pictures, And I'm sure most of you would have answers to these. So do you recognize these animals? I bet you got them all right. In fact, I'm sure that some of you are so smart that you even know the binomial names of these. And I profess that I don't remember binomial names very well. Okay, now let me show you another set. Do you know that these are animals too? The previous set that I showed you, you might find them in zoos, or you might have to go to distant places to see them. These are all around us. I'm sure some of you may recognize some of these, but in general, these are not very well known. For example, the face mite, Demodex, is present on almost all adults. It lives its entire life on the human skin. And it has evolved along with the human skin for a few million years. And we know nothing about it. Many of these pictures that you see are inhabitants of soil and pond systems that even if you perhaps take the effort to walk 20 steps ahead of you and pick up some soil or pick up a little bit of pond water, you're likely to see them. These indeed are our poorly studied microscopic cousins. Now let me show you this video. I'm sure many of you have seen videos like this on the Discovery Channel, perhaps read about it or been fascinated by it as I was as a child. And I'm going to sort of take you to this. So here you have a cheetah, I call him Ramu the cheetah, who's hungry. So he chases a gazelle down, Shamu the gazelle, and he hunts him down, and then in due course, gets his lunch. Now, in order for you to see this, you need to go to Africa. You need to actually hope that the weather is good. You find a cheetah, you find a gazelle, you find them at the same time, and you are able to see a cheetah hunt a gazelle. Let me show you another video. Okay. So now you're seeing what I call Mr. Vorax. If you take this is a single-celled organism that is found commonly in pond water. You take a single cell of Mr. Vorax and let him grow in the pond water. Normally, Mr. Vorax is tear-shaped like this, or a pearl-shaped organism like this. As he grows in the pond water, he's enjoying himself, he has his food. At some point, the resources in his system go down. And when that happens, interestingly, a small population of him or his kin become giants, almost 7 to 8 times their size. And then, strangely, these giants eat these small ones, a form of cannibalism, right? So here is here is a pearl-shaped one, and here's a giant actually, and you see these blue spots. They are actually contractile vacuoles containing borax that have been consumed by this giant. Okay, now the third type that is there, you see, is this borax that has developed a tail. The interesting thing is that this Mr. Vorax cannot be eaten by the giant and is resistant to being eaten. So in a population of Tetrahymenovorax, you have 3 different types that come up. The pearl-shaped one, or that is one type, the giant, and the tailed one, all of which are in a rock, paper, scissors type of equilibrium. What it means is you must have played the game rock, paper, and scissors. No one of rock, paper, or scissors is better than both the others. So, for example, the scissors can be broken by the rock, the paper can be cut by the scissors, and the rock can be covered by the paper. So there's no one which is better. Now, this is something that even if you spend a little effort, perhaps a couple of weeks, you can find this organism. You'll be able to study this phenomenon right in the precincts of your own house. Don't you think this is a far more interesting story, or equally interesting story, as the gazelle and the cheetah? So the point that I'm trying to make is that we barely know 90% or perhaps 99% of life around us. There was a study that said that actually tried to survey about the species that are on Earth. And what it essentially said was that if we carefully catalog every species that is there on Earth, it will take us about 480 years to completely comprehensively catalog them. Now, it made a very, very simple back of the envelope assumption that there are about 11,000 bacteria and archaea. Now, what we are now realizing over time, and this study was published 10 years ago, is that there are at least 10 to 100 million different species of microscopic bacteria, microscopic forms that we call prokaryotes. And in order for us to study them, as you can see, a simple multiplication will show you that it is going to take 1,000 human lifetimes to be able to achieve that. Further, most of that life form is microscopic. So, having said all this, the bottom line is we do not see 99% of life around us. For a while, this was so in the history of humans. And there were speculations in various older texts that there is small life that we don't see and the like. But it required a big invention of the microscope for us to have access to that microbial life. And this is the first type of scientist that you often see, which is the inventor. Now, the inventor is essentially a person who makes new devices or methods that eases scientific investigations. The history of microscopy is somewhat murky, and Zacharias Janssen is the first person to have invented the microscope. But in reality, his contributions towards microbial life is non-existent. So often that is the state of the inventor. He may not exactly be involved in using his instrument greatly, but nevertheless, he is an inventor and is a very important spoke in the sciences. Microscopy has had many, many different inventions, and all of them have been greatly lauded in the community of scientists, because from seeing single cells, we are now able to look at protein complexes. We are able to go down to the very basis. We are able to visualize the very basis of life. And hence, these are a very great set of lauded people. As you can see, that many of them have been recipients of the famed Nobel Prize. But an instrument by itself is of little use until you have a user. So, although Zacharias Janssen perhaps invented his compound microscope in the 1500s, it took a Dutch trader, Antony van Leeuwenhoek, to actually use the microscope. Now, this is a very interesting story. Andre van Leeuwenhoek was a cloth trader. He had no formal education in the sciences, but he was an extraordinarily curious person. And this is the second kind of person that is very important in the sciences, which is an explorer. So the explorer is a person who makes foundational observations or simply creates a catalog of observations as he explores new frontiers of the sciences. So Leeuwenhoek initially perhaps made the microscope to look at his clothes that he was weaving. But then he put the microscope onto other aspects. And in fact, he was the first one to describe life forms that people had no idea about. For example, over here you see a picture with his descriptions of bacteria that he saw, or life forms, or animalcules, as he called them, that he saw in the teeth, in his own teeth. Here are his observations of the vinegar eel, which spoils vinegar. And this is the little microscopic animal that spoils it. And then all other observations and all other things that all other life forms that are there in his vinegar eel, and so on. So he made a large catalog of observations, and he used to write them in Dutch and send them to the Royal Society of England, and they were regaled by his observations. And in fact, his observations inspired a whole generation of scientists to take this up more seriously and study this microbial life. The greatest thing about this, the most inspiring thing about this, was that he had no formal education, but he used his intuition and his passion and interest to pursue his hobby of looking at microbial life and made some astounding discoveries. All right, let me now show you another video. Now, what we see here is an organism called Diphalgia and another one called Spirogyra. Diphalgia eats Spirogyra for a living. So what I want you to pay your attention on is this is the Diphalgia. It's at the end of a Spirogyra. Look what happens. You can see that it sucks up the chloroplast and has a nice meal, a nice slurp, just as we have a good sip of soda or more or whatever that you like to have with a straw. First person who studied this was an extraordinary scientist by the name of Joseph Leidy. Now, Joseph Leidy was a dinosaur biologist. He was the first one to actually get a full fossil of a dinosaur. And he was a very great dinosaur scientist. At the age of 50, he got a microscope as a gift. And then he started looking at microscopic life. And some of his drawings, as you can see, are way better than what we can even expect to see in a textbook today because of his attention for details. And in fact, there's a book based on his life, which is titled The Last Man Who Knew Everything. Joseph Leidy was a remarkable man. He introduced us to a variety of microscopic life, and he cataloged them just as many of these explorers are supposed to do. And his writings are still available today for us to see. A couple of other things about this man. He was one of the first ones to use the microscope to solve a murder mystery. He was the first one to show that the blood on the person who was alleged to have done the murder was actually human blood and not sheep blood, as he had said. By the 1800s, microscopy came out to be more popular. In fact, here's an image where you see a Victorian lady with her microscope next to her. Here's a caricature of the River Thames, which was so polluted, and they had no way to actually tell people. So they had this extraordinary cartoon where this woman says that she's looking at this monster soup in the River Thames. So microscopy had a phase in the 1800s where it became more commonly available of course, to people who could afford the microscope. But in terms of the sciences, there was still more progress. Let me show you a couple of other videos. Over here, what you're seeing are yeast cells. These yeast cells, these yeast cells were put in some sugar solution, and this is an advancing bubble of carbon dioxide that is pushing the yeast cells aside. Fascinating. In fact, yeast, as you know, is used to make bread. Yeast is also used to make wine. But for a long period, people thought that the conversion of a fruit juice to a wine was a natural process, a physiological process that happens to fruit juice. Here is something that might be familiar to you. This is a batter of idli. And in the idli batter, you see these bubbles that are forming. In fact, these are the bubbles that make the idli a very delectable and soft dish. This is a process that is also a fermentation process done by various bacteria. The man who actually studied this, or who was the first one to reveal the importance of fermentation, was a giant in the field of microbiology. Louis Pasteur is what I would call a conceptualist. Now, a conceptualist is a person who produces a new theory or discovers a new law that explains a large body of previous observations. Now, everybody knew fermentation for thousands of years, and they thought it was a natural process. But it was Louis Pasteur who said that actually it's the yeast in that grape juice that ferments, that converts that sugar into an alcohol. Louis Pasteur was an amazing scientist. He was involved in many different first experiments. So, for example, he was the first one to refute spontaneous generation. In the old days, it was thought that life actually can arise from soil spontaneously, but his experiment showed no, that it is not so. You need life to beget life. And he had many other things that still impact our lives today. For example, the process of pasteurization to preserve milk, wines, and many other food items was something that he had conceived when he studied microbes. Most interestingly, he was also one of the fathers of the germ theory of diseases. And interestingly, very important in the history of vaccination. So I'm not going to spend too much time on Louis Pasteur, except to show you a great experiment that he did, which is quite relevant even today. So chicken cholera was a big veterinary problem. And Louis Pasteur realized that you can actually grow the bacterium that causes chicken cholera in a flask with a culture medium that he had devised. And if you take a bit of that culture medium and inject the chicken, it is likely to die because of the cholera. So he's able to show that indeed this bacterium is the cause for this. Now, it so happened that he had asked his assistant to inject the healthy chicken, and his assistant forgot about it. After a month, Pasteur thought, well, what the hell, I'll inject the chicken anyway. So he injected the healthy chicken with this aged culture of the cholera bacterium. The chicken survived. After a few weeks, he again injected it with a fresh culture of bacterium that he knew would kill the chicken. But the one that was inoculated first with the aged culture survived. And this opened up a whole new world and whole new set of ideas about how, if you know what causes a disease, you can attenuate the organism that causes the disease. You can make it less potent in some way, maybe by just aging it, letting it be, not giving it too many nutrients, one of many ways, such that when it is injected, it would actually provide resistance to the disease rather than you succumbing to the disease. So Pasteur was the first one to demonstrate this. He was also a very big showman. And in 1881, in the month of May, he said that he has a cure for a terrible disease called anthracis, the anthrax disease that infected a large number of veterinary animals. So he called a big assembly, called all the farmers from around the place and all the scientists. And there were a lot of skeptics at this time. So this was the famous experiment at Puy-le-Fort in France. There, what he did was he did the same experiment as he did with the chicken, in which he had 60 farm animals, mostly sheep. He divided them into two groups of 25 each. And then in one, he did an experiment. He injected them with an aged culture or some attenuated culture of Bacillus anthracis. And after 2 weeks, he then again, or after a few weeks, he again re-injected them with a potent culture of Bacillus anthracis. Those that received the attenuated culture, or the vaccine as we call it, all those animals survived, whereas those that didn't receive the vaccine died. So this opened up a whole new field. This demonstration was not just for scientists who understand this, but for all the farmers around them. And this is how, by example, Pasteur showed that some things like vaccination work. And this inspired a whole generation of people to bring out vaccines. Even what we see today has some primer in this primary experiment of Louis Pasteur. But science also progresses by what I call as a technician. Technician is a person who performs technically demanding tasks. His use of his hands and his eyes is very important, and he has long hours of focus and concentration to achieve this. The technician is one of the most important people to the sciences. Robert Koch was a remarkable bacteriologist. All the techniques that you will learn in a microbiology class, which is using a Petri dish, using agar-agar, and so on and so forth, all came from inventions from his group. Interestingly, like Joseph Leidy, Robert Koch was also got a gift a bit earlier in his life of a microscope, and he converted a home lab, a small part of his home, into a lab, and then began his investigations into microbial life. Now, he's indeed one of the fathers of modern medical microbiology. He discovered the bacteria that cause anthrax, tuberculosis, and cholera. And he's very famous for his Koch's postulates that a microorganism that is suspected to cause a disease must be found in the diseased individual, must be cultured from the diseased individual. When you take that and re-inoculate a healthy individual, it should cause the disease. And then you should be able to re-isolate the microbe. So this Koch's postulate is the base, the bedrock of proving that an organism causes a disease. And this is not just so for animal diseases, but also in plants and even, for that matter, microbial life. All right, so I shall proceed. I shall skip a few videos. So a lot of medical microbiology today has its antecedents in these people who explored microbial life. But if you were to think, or if I'm sort of implying that you all should become germ phobes and should be sanitizing yourself or bathing in the hand sanitizers that is found all over today, remember this fact. A human being perhaps has about, I would say about 10 trillion cells, or maybe 30 trillion cells in his body. The number of bacteria, the number of microbial life that is on us is about 100 trillion cells. So this human being that is speaking today, this animal in me is just 10% of the cells. 90% of the cells are bacteria of different kind. And this is true for each one of you who's listening too. So it's not that we are not without them. They are actually part of they coat us. They are all over our skin. They are in our gut. And in fact, many of them have got protective roles and provide things such as protections and vitamins and so on that we cannot make ourselves. Okay, so moving on from medical microbiology, I'm sure in school, for those who are in school, you have learned about the famed energy pyramid, primary producers, consumers, secondary consumers, and the like. Now, when you think about primary producers, you're then taught about photosynthesis, and organisms acquire their energy from sunlight, we are taught, and materials from non-living sources, and use carbon dioxide and release oxygen, as shown in this very simple cartoon. Sunlight, a plant uses carbon dioxide to photosynthesize, to fix carbon, and in the process releases oxygen. So when you think about photosynthesis, perhaps you have an image of this beautiful place. This is, I think, somewhere near Ooty that I took this picture. But in reality, the picture is very different. If you look at the Earth and you look at all these green zones, this is where photosynthesis occurs maximally. This is a satellite picture. And what you see is that, yes, there are these green patches on terrestrial parts of Earth, but the marine life is greatly responsible for a large amount of photosynthesis, which means the marine life is very important for the amount of oxygen that is there in the air and that we breathe in. In fact, there has been a calculation that at least 50% of all the oxygen that we breathe come because of microscopic organisms that are in the marine environments, even in ponds, in rivers, and also the great oceans. So here are some interesting videos which I've taken, and I shall talk about this later. This is Micrasterius, which is two cells that is about to divide. And you see here, this is one large chloroplast. Very interesting. And here's Merismopedia, which is a cyanobacterium. It has a very interesting form of movement, as you can see, a very interesting form of gliding motion. Instead of looking at the tree in Ooty, when I think of photosynthesis, I think of all these other denizens whom we don't see and whom we have an opportunity to see or study, and about whom very little is known. All right, so let's move on. When you think about farming, and I'm sure many of you had these lessons in farming, and I know I failed this when I was in my 7th grade, so I'm sort of revising it. When you think about farming, there is this understanding now that farming began in humans about 11,000 years ago, or maybe 13,000 years ago in the Fertile Crescent. And there were many, many places where farming began. This is just about 11,000 years old. And this has been critical for the development of human cities and human societies. But did you know that 50 million years ago, an ant called the leafcutter ant began farming of fungi? So what happens is that the leafcutter ant nicely is known as a leafcutter ant because it has this very interesting way of cutting leaves. It brings the leaf back to its nest and uses it to grow fungi, and the fungi is its primary source of nutrition. And it is very meticulous about ensuring that the culture is very carefully maintained. It is very meticulous about ensuring that it doesn't get contaminated by other fungi. So we are not the first farmers. There are life forms that have come before us. In fact, the even more spectacular example of farming is this amoeba called Dictyostelium. It turns out that it always carries with it a set of bacteria that, as you can see, as these two dots, that when it disperses its spores, it carries its bacteria along with it and sets up a little farm wherever it is. Dictyostelia are social amoeba. They're very interesting. What happens is that they are singular, they're unicellular rather, generally, but when there is food shortage, they get together and form this thing called as a fruiting body. And then when conditions are favorable, the fruiting body breaks apart and are dispersed. So in this process, Dictyostelium also holds on to some of its bacterial seeds, so to say, so that it can, wherever it is dispersed, the bacteria grow again, and then Dictyostelium has its own set, has its own source for food. So we are not the first farmers. Microbial life had figured this out way, way, way before humans were there on Earth. Now here's a more dramatic example of the same. This little green organism you see here, it's an organism called Paramecium bursaria. All these others that you see around are other Paramecia. Now notice that Paramecium bursaria has got these green bodies inside. These are actually a completely different organism called a chlorophyte alga. They're called zoochlorellae. And this Paramecium bursaria basically doesn't have to worry about food. Whatever food it needs, it already gets from the carbon dioxide that is fixed into various carbon molecules, in various organic molecules by these zoochlorella. So essentially, you're carrying your food within yourself. I find this fascinating because this culture of Paramecium, this green Paramecium bursaria, I found in a little pond about 2 years ago. And if I take you to my home lab today, it's still thriving. Indeed, because it doesn't have to worry about its food source anymore. It's there inside it. Fascinating, isn't it? So we are not the first farmers. There are many more inspiring lives and organisms around us. Okay, now a lot of observations have been made on microbial life using the microscope, and lots of scientists have contributed to understanding the biology of this. But yet, as I told you, it's going to take us a few thousand generations of humans, or maybe 1,000 lifetimes, for us to figure out the story of each and every life form. Now, in the field of microbiology, or in the field of biology per se, one of the biggest stages in its history was the discovery that DNA, which is a hereditary material, has a double helical structure. And this is another type of scientist that fascinates me, and I call him the ontologist. The ontologist is a person who finds order in data and categorizes his observations and finds relationships between categories. He's able to combine disparate fields of knowledge and place them in a unified framework. This unification of the structure of DNA by Francis Crick and Jim Watson was monumental, because now we knew the basis for hereditary. We knew why like begets like and how like begets like. From this discovery of the structure of DNA came many ideas on the molecular biology of life. So we now have what we call as a central dogma of biology, where we know that you can get DNA from DNA by a set of proteins that are called replication enzymes. And then the flow of information. So most of our body, most of our work is done by proteins. So we now know the flow of information. That is, how do you get a gene which is there in DNA to enact itself? So you have transcription to make RNA, transcription as a set of molecules to make RNA, and then you have translation that makes protein. So replication, transcription, and translation are the central flow of information, is the highway of the central flow of information of life. Now, this is perhaps a little much for some of you, but the important thing that came out is that when we started looking for enzymes that are involved, say, for example, in translation across life, when we look for enzymes that are involved in translation and transcription across humans, plants, bacteria, archaea, microscopic life, the thing that's shown through all this is that the code is very similar. What it meant, that all life has a common origin. In fact, anything that is living around us is in a sense related to us. And in fact, this is a very profound insight about the history and the evolution of life that I shall not dwell on, but that is where most of my current work is. But then the fact that you have information in DNA led to another big and fascinating story, which is the whole basis for recombinant DNA technology. And why I bring this up is because in microbes are also in conflict with other microbes and also with viruses that attack microbes. So the viruses that attack a bacterium are called bacteriophages. And what happens is that the bacterium also wants to fight back when it is being attacked by the bacteriophage. And what happens is bacterium injects its DNA, and then there's this system called as a restriction system that cuts this bacterial DNA. And then there's this modification system that modifies the host DNA to make sure that this toxin, this restriction enzyme, does not cut its own DNA, but only cuts foreign DNA. So humans are pretty clever at using this to make technologies. And what happened was that this is the basis for the recombinant DNA technology, because now you know that if you can get a restriction enzyme that cuts DNA, you can take a piece of DNA from any source and put it in and attach it using all those enzymes that you've studied, that you've understood, that are involved in replication and transcription, and put together a new molecule that can have very interesting concept. I shall not dwell on this, but this has been a very big contributor to our understanding of life, our understanding of the way microbes work, but also to technologies that have revolutionized our understanding. Now, coming much further, in 1995, I was a student, a PhD student, working in a lab when I first saw this paper of the first sequencing of the human genome. The Haemophilus influenza genome. This is a little bacterium. And in those days, when we were students, we never thought that we will have the entire genetic information of an organism. And it was achieved. It was a technological innovation, again, by one of the scientists who was involved in the history of developing recombinant DNA technology, and enterprising scientist by the name Craig Venter. And with this, they invented a technology by which you can get the sequence of all the DNA of an organism. At that point, I thought, if I am crazy about studying information, then I need to be able to understand all information if possible. And over time, thousands of genomes have been sequenced, and many great insights about life have come about, because now, when you have all information about a large diverse set of life forms, you can actually now ask the question, where did this come from? I'm not going to dwell on this, but it has helped us answer the origins of life to a very great degree of finesse today. Interestingly, when you study this, you realize that most of life actually is unicellular, as I said. And you have two forms of life. One, those have nucleus, and those that don't. And I used to think that nucleus and life forms that have a nucleus are large and macroscopic. But in fact, I realize, and we see now, that most of life, whether it has a nucleus or not, is microscopic. So that's what it is. So the advantage of having so much genome, so much information, this whole genome technology and all information about life, is that now you can go in the reverse direction. You study these genes on a computer, and then you can ask the question, can I find new things? And this is the frontier of biology today, where you're using information to go back and study the organism. Here's a very small example, and I shall stop. This is perhaps my last example today. This is the oral cavity of a human. Normally, there are about 200 species of bacteria in our mouths, and no two mouths are identical. That is, the number of species, the types of species that are there in one person's mouth is very different from that of another person's mouth. So one of the big questions that we had in the lab that I work in is that If that is so, then how do bacteria know each other? That is, how does a bacterium know that this other bacterium that is touching me actually is similar to me or not? So this was done entirely computationally. And what we realized was that bacteria recognize each other by a very interesting mechanism. So you have bacteria that you may call them as having spears, and they have a poison-tipped spear over here. And when two bacteria meet together, when two bacteria come in contact with each other, they actually spear each other with these poison tips. And if you are of the same type, you have an antidote to this poison tip. But if you are different, then you do not have the antidote to this poison tip. And then one of them, the one that is being speared, the bacteria that is being speared, is killed. So this was entirely predicted computationally by studying the genes and arriving at a hypothesis that, hey, this is something that we think is very common in bacterial life. And then when experiments were done in various bacterial systems, it confirmed this idea. In fact, not only did we predict that this is done by sticks, there's another way whereby bacteria shoot out toxins into the medium. Like, say, you may think of it like bullets. And when they shoot out these bullets, if the two bacteria are of the same type, they have an antidote to the bullet, so they don't kill each other. But if they are different, then it's a healthy competition, and the one with the better bullet wins. So microbial life is fascinating. This came entirely from computational predictions, but there's so much more out there that we have very little idea about yet. And because we now have a lot of information, we can go back and forth from experiment to theory and come up with a unified view of microbial life. All right, so I have spoken much, and I just want to end with a small blurb, and that I spoke about microbial life and the importance of microscopes. Microscopes are heavy, expensive. Most people don't even have access to one. So how are we going to study microbial life? How are we going to study 99% of life around us when we don't even have the tools to see it? And only those scientists and rich people and people with access to these have. And that's how it was for, say, until the year 2000 or 2000, maybe 14 or 15. Around 2015, there was this remarkable scientist at Stanford University, Manu Prakash, and his student Jim Zibulski, who came up with a remarkable invention called as the foldscope. Now, the foldscope is basically based on a paper frame. It is called as an origami microscope, and it has a single ball lens, just as Leeuwenhoek had ball lenses. And it can magnify whatever you see to 140 times. All the videos that I showed you today, all the microscopic pictures that I showed you today, they were done in the basement of my house, and they were taken using this foldscope over here, or a foldscope very much like this. The foldscope, the original idea was that it would cost 50 cents or $1, maybe about 70 rupees. It's widely available across the world. It's widely available in India. The Department of Biotechnology in India collaborated with the Prakash Lab and with the foldscope team and have disseminated it in villages of India. The great thing about this is you can write your observations in a common website, as the website is up here. And what I find most fascinating is that one of the things that has always bothered me is that if you want to do science, you have to know English. But here is a forum where you can describe your observations in the language of your choice. So, for example, one of the the greatest, you may say, foldscope user is a person who lives in Madurai. He runs a small school, and these are his observations written entirely in Tamil, and they are fascinating. And he has conducted several camps across Madurai and Tamil Nadu to help teachers and students understand how to use the foldscope and access that microbial life that I've been talking about in my talk today. So We'll never get a count for how many species that are out there. But if you join this, if you have an access to an instrument such as a foldscope, you can be a part of this community. You can be a part of this. And as I said, Leeuwenhoek didn't know what was a microscopic life around him. He just went ahead and saw. And that's the beauty of having such an instrument with yourself. So I will end with a quote by Dr. E.O. Wilson, who's a very famous biologist. And he said that if I could live my life all over again, I would be a microbial ecologist. 10 billion bacteria living in a gram of ordinary soil, a mere pinch held between the thumb and forefingers. They represent thousands of species, none of which are known to science. Into that world, I would go with the aid of a modern microscope and molecular tools, and I would cut my way through the clonal forest, sprawled across grains of sand, travel in an imagined submarine through drops of water, proportionately the size of lakes, and track predators and prey in order to discover new lifeways and alien footbeds. We are at this crucial juncture where we need more hands to join forces to study this microbial world that is still unknown to most of us. Now, finally, this is an observation that I made in a pond. This is actually a pond leaf. And you can see that there are these organisms that have this interesting motion that they are talking to each other. Maybe they're discussing about what's happening there, or they're exchanging information, and then they fly away. Nobody knows what this is. This is an observation made in an ordinary pond that is found very close to me. My friend Manu Prakash calls it synchronous disaggregation. What is it? What is the biology? What is the basis of this? We don't know. There are so many such mysteries waiting to be discovered. And I hope that you all, or some of you, become part of this process of discovery. So thank you for patiently listening to me. And for most of you, you have a lifetime of learning ahead of you.
Fantastic. Dr. Lakshminarayan?
Yes.
Could you remove the share screen feature so that we can see you? Yeah. Phenomenal. Again, I'm amazed by what you have shared with us today. Unbelievable. I'm very honored to introduce my I would say guru. I'm so honored that Dr. Sivadanu Pillai is with us today. Dr. Pillai, I would like to listen to you for a few minutes. Please unmute. Kindly unmute and share a few words with us, sir.
Sir, it was a fantastic lecture. Many things which I didn't know, I have understood today. So this is a word is, phenomenon. Thanks to Mr. Iyer for giving a wonderful speech. It's not a speech, it's a research what he has done. So thanks to you, Mr. Iyer.
Lakshminarayan sir, earlier I was telling you, I am honored to introduce you to Dr. Sivadanu Pillai, who I consider the premier scientist in rocket and missile technology. I'm so honored, Dr. Pillai, you are with us today.
It's a great honor to know you.
Thank you, sir. All your programs are wonderful, sir. Lot of learning. Yes. We think that we have learned. No, we have not learned. Only the tip.
Yeah, I think in your lecture you said very nicely, கற்றது கைமண்ணளவு கல்லாது உலகளவுன்னு சொல்லிட்டு நீங்க ரொம்ப அருமையா சொன்னீங்க. So, Velmurugan, I would like to introduce you to our friend Dr. Lakshminarayan Iyer. Velmurugan is listening to you from Singapore. Please unmute Velmurugan. And he is also part of our Visa Foundation. He is the regional coordinator there. Velmurugan, could you please unmute and say hello? We're not able to hear you, Velmurugan. You are still, you have to unmute, please. Kindly unmute. Looks like he's not able to. Can you unmute Velmurugan? We still are not able to listen to you. Your speaker is. Dr. Ardra Kachru?
I'm here.
Okay, you can start the question answer program. I see quite a few children are already ready to ask some questions. You can go ahead
Yes.
and please go
Yes,
on.
I just want to say, Lakshmi, what a wonderful, wonderful talk. It reminded me of two things. One, it took me back to the first day in microbiology lab and BSc. That's when I first met Dr. Iyer, and we were lab partners. So thanks for bringing back, that microbial world to me, and thanks for connecting the history and medicine, agriculture, and all other aspects of life and putting it all together. The other thing that it reminded me of is that we call Dr. Iyer, some friends call him Dumbo, because he seems to know everything about everything. And that was personified in this talk. He just covered, I can't even count the number of topics that he covered in a 50-minute talk. So just amazing. So I'm happy to take questions. I see some raised hands. Some of you don't have your names on there, so I'm unable to call you out by name. Just please unmute yourself and ask the question, or you're welcome to type the question and I will read it out. So there's somebody on Android, Blue Droid, that has their hand up. Would you like to ask your question?
Please unmute and then ask your question, then mute again after the question. Are there any questions in the chat box in the meantime?
Yeah, I don't see any yet, but maybe we can give people some time to type. Also, while you're thinking of questions, I can vouch for the Foldscope. I purchased one because Dr. Iyer introduced me to it, and I can't tell you, it's an amazing tool, and it's cheap, it's affordable. If you can, I urge you to get one. It's just fascinating.
Right.
Lakshmana
And the
is
best thing,
here.
the best thing I like about the Foldscope is the community. The community that actually interacts is a mixture of scientists, painters, artists, school children. The youngest post is a post by a 5-year-old. The oldest one, I don't know, nobody tells their age when they're old. So you have a fantastic group of people interacting. And the great thing I like about it is that it connects Madurai to South Africa, because you have people making observations, children making observations in a place like South Africa, children making observations in a place like Madurai, and exchanging information in a common platform. I find that fascinating, and I encourage you all to explore this in your various groups that you all work in. The Foldscope is greatly supported by the government of India and widely used in this pandemic era to actually help children explore the microbial world.
Dr. Lakshminarayan, you have given me a wonderful idea. We conduct quite a few scientific experiments, encouraging children to conduct on their own and then have exhibits and all that. We did one for 48 schools in Tiruvallur district, and 7,750 students had participated. And then we give them honor them with awards and all that. So from now on, I'll probably procure these Foldscopes and give them as part of the gift. We give a set of books to them. And that way we will encourage them to become also microbiologists and explorers and innovators, as you rightly described. So we will do that. From now on, we will make sure children have access to these. And we will start. You've given me a fantastic idea today. Dr. Pillai sir, can you give me any other ideas? What else we can do to promote this Foldscope to reach children? Can you give me some ideas? What else I can do?
But I was only thinking, not the idea. I was thinking about the corona, that how they have, anyway, it is related to what Dr. Iyer has explained. The scientists who are involved in the bio warfare, how do they use animal to take out and then form this bio. Dr. Iyer, what is the theory behind that?
This is fairly controversial, because when we work with pathogens, sometimes the pathogens might escape a lab. And one of the idea that is floating around is that people who were studying these viruses were a bit careless, and it came out from the lab. But I want to tell you something more interesting. Biowarfare, we think, was first used by humans. I'm sorry to say no. If you ever study a wasp that actually neutralizes a spider, and very common in India, I enjoyed studying wasps and spiders. They actually inject a virus into the spider so that the virus neutralizes the wasp. The wasp neutralizes the spider, and the spider does not move, and the wasp can take the spider to its nest, and it's a source of food. So biowarfare is something that has been used maybe a few million years before even humans conceived that idea.
Fascinating. Really fascinating, isn't it, Melissa? Didn't know that. My
Yeah,
God.
Vijay sir,
okay.
I think I'm able to.
Where go ahead. Yeah, please. We want to.
Yeah, sir, Iyer sir and Sotanu sir. Good evening and good morning from Singapore. I'm very glad that to see you. I know from day one with Dr. Kalam tour with India and Singapore. Sotanu sir, how much he closely associated with Dr. Kalam and empowering youths. Last few sessions, I seen the record from Sivadanu Pillai sir as well. So thanks for that. The great the missile technology and everything sharing with our small people is really, really amazing, sir. Thanks from Singapore, and then same to Iyer also. One question to Iyer sir, that I'm working in pharmaceutical last 25 years. So I also is working in microbiology, and then normal drugs and everything like acute roda and some other thing. But quite interesting that you're telling that how the behaviors and everything. My mind is asking only one question, that how you validate it, and then how you can sequence all those kind of things, making it quite theory and hypothesis, and then the validation. How long it takes in time, how making such a kind of certainty you can making that yes, this is the behavior is really, really very, very hard, because one small drug is a normal drug, also developing that in the solid dose, we taking 10 years, 15 years, something like that. How much is complex on that? I can visualize it. Maybe can give some kind of the idea how you're doing and validate all this kind of process, sir.
I can give you an analogy. Think about astronomy. For the longest period of time, people observed the sky and wondered, what are these things that are moving in particular patterns or blinking in particular ways? It was only when we applied the telescope and actually a large number of observations, careful catalogs of the night sky, variable stars, open clusters, galaxies, that we began to understand the dynamics of how stars evolve or how stars come about. So there's a constant accumulation of knowledge. And when you have a sufficient body of knowledge that is fairly diverse, the theoretician comes in and says, okay, now I'm looking at all these stars. Is there a pattern that can explain the evolution of stars? So there's a very famous diagram in astronomy called as the Hertzsprung-Russell diagram, which actually explains how a star evolves. So now if you go to a astronomy book, they'll say, oh, a star starts off as a certain size, and then it consumes a lot of fuel and becomes big, and then depending upon its mass, it can either become a black hole or a white dwarf or whatever. All that comes because of a large number of observations. Similarly, in biology, we have reached a point where we have not only a large number of observations, we have the genetic basis, information of life. So combining all of this is now becoming easier, because we now have a large number of people who have made databases, for example, by which we can study these things. So you can probe databases, asking directed as well as undirected questions. It's almost like this. I look at myself as a researcher, as a person who goes to a forest. I go into a forest trying to look for a deer, and then I find this very interesting iridescent beetle, and that catches my attention. And there I am looking at the beetle, and I make a new discovery that, hey, this beetle actually rears fungi, and which I find, as I said, was very fascinating. So that's how the nature of knowledge is, that as when you categorize it and keep it in a way which is accessible to people, it's open to discovery. And we've reached that point in biology where now theory can feed back into experiments. And this was so in physics. Had it not been for a large body of observations in astronomy, we would have never made all these great theories about physics. We would never know the planetary motion of of our planets. We would not know that what type of motion they have had it not been for the great observations of Tycho Brahe and Kepler and so on. So that's how knowledge is. It reaches a point where now theory can feed back into experiment. And of course, experiment feeds back into theory. So we are at that critical point in biology, and I'm really excited for the next 50 to 100 years, because I think we are going to rewrite our textbooks based on what we understand.
Fantastic. Dr. Pillai, sir.
So I have a question in chat first. So
Okay.
is
Yeah,
that
go
okay if I?
Yeah, that's fine. Yeah.
Yeah, Murugan Kalaipan asks, "Hi sir, how can we introduce such microbiological studies in Indian school levels?"
So, if you ask me, the one that has been very successful so far has been using the foldscope. There are local foldscope clubs in India. For example, there's one in a village in Madurai, and there are many people who go around India educating people about how to use the paper microscope. The paper microscope, there's even a DBT grant which you can write about, write to DBT. They'll provide you some money and some foldscopes so you can explore the foldscope in your school setting. The best way to explore microbial life is to let the child explore it. Any directed study is good. Directed study is good, but biology is about being outside, looking at life around you. And there's nothing like giving a microscope to a kid and saying, go do whatever you want, jump on it, it's not going to break. Tear it, don't worry about it. Normally when I was a student in my school, we had one microscope and 50 kids peering into it. The teacher would say, I'm now going to show you an onion cell. And you have exactly 5 minutes to see this, and don't touch the microscope. And then we peer in, and it would be totally out of focus. And some of us would have specs and would not know what we are looking at. And then we'd look at our textbook and say, oh, I see this. Basically, you're not seeing anything. The greatest thing about using your own device without fear of it breaking is that you make the observations. So try and get a bunch of foldscope in your schools. Maybe get 10 of them, give it to the kids, train them a little bit. It takes 1 day to train them, and say, go explore the world around you. You will be surprised as to what they discover.
We will implement it, sir. I promise you that. Dr. Pillai, sir, how do we make that kind of observation from children appealing to the education boards in India? Is there a way to approach them?
Sir, in our preliminary, we are killing the enthusiasm of the children, and we kill the creativity. That's what is going on in India. What Dr. Iyer is saying, you give it to the child, see, the child will explore. That is not going to happen in India with the present system. So we have to completely change our education system, make the children free. They should think free. They should have the creativity to know many things. Then it helps. One simple example I want to tell you. My grandson, he was in US. When he was studying in the first grade, he was taken to the fire engine. So the teacher explains the whole fire engine, but these people, they interact with the firemen and they conduct a fire show, actually how it should be done, everything they show. And then the children are allowed to operate the fire engine. What age? I'm talking about age 6. They do the complete fire, removing the fire, everything they do. That grandson now is 20 years. If I ask him about fire engine, every part of fire engine he tells. This is the way of education. It is not there in India. So I was telling this minister recently, there was a deliberation with the Tamil Nadu education minister, primary education minister. So I told him, what is the culture in the other places? In places like Denmark and other places, they allow the child to go to school after 7 years. Here, now the new policy which has come, the 3rd year onwards, 3rd year, age 3, the 3 years before first standard, they have made it compulsory now. the new
Oh,
education system.
huh.
And then fifth standard, there's a board examination.
Oh my.
What is board examination? You have to study as it is in the book and write.
Oh,
Completely, they want to kill the creativity. That's
oh.
what is happening here. What Dr. Iyer was telling you, buy a microscope, then tell them one day, then next day onwards, they will find out. True. That is what is education. Here it is not there.
Yes, and I just want to interject for a second that we did this experiment. This is a kind of social experiment, you may say, in India, in Madurai. There's this foundation called the Eden Educational Foundation, run by an idealist teacher, and the students basically could not afford the microscope. We sent him microscopes. We taught him how to, Pandya Rajan is his name. He did this remarkable exercise where he not only trained his kids, they trained other kids. And in about 2 years, they went to colleges to make demonstrations about how to use or how they could use the paper microscope, and describe what is inside it. So I think it is possible. It requires a lot of good souls, a lot of, people to get through, but the tool is cheap enough that you don't have to worry about it breaking or, kids spoiling it. They can actually take it home for a week, check it, and then enjoy.
10th, 11th, Dr. Iyer, even 10th, 11th standard, we are not allowed to touch the microscope.
Correct,
Oh,
correct. I know, I experienced that, yeah.
Pillai sir, here, the NASA is conducting once a year from age 5 to 9, 1 week space camp. I was amazed to hear that. Age 5 to 9, they conduct this space camp. And then high school students, as you rightly said, here are already, I'm talking about at least from 15, 20 years back, they were already developing CubeSats. That amazed me. And then while in high school, you are linked with one of the college professors in the local colleges, and you do some experiments and submit a paper before you graduate your high school. So I think this society somehow figured out that children should be given the opportunity to innovate, create, and I think that's one of the nice things.
Dr. Kalam was always talking about this.
Yes.
In the early education, how you induce creativity to the children. He always talked about it.
Exactly.
Now, one thing is like this Madurai model. Can we create some model? Now, since we are taking lead in developing children, suppose we create some society. Create pockets of excellence. Then it will speak that, the creativity, how we can introduce.
Fantastic. Yeah. Children who are listening to us today, the youngsters who are in the audience today, if you write a 2-page note and email it to me, we will select 5 of them, and the 5 who are selected will receive this whole scope as a gift. We'll start with that. How about it, Dr. Kachru? Can we do that?
That's wonderful. I'm very happy to read if they send the write-up. That would be
Dr. Kachru and Dr. Rajan, Gayatri Rajan, our trustees will review the things. Those who are participating today. All right, so just a 2-page thing at what you observed today and what you learned, and what you want to do with that information you learned from Dr. Lakshmi Narayan and Dr. Sivanand Pillai. Write to us and we'll select 5 of those, and each of you will get the full scope as a gift. Anthony Raj, I see your hand raising. Dr. Kachru, can we ask them to?
Yeah, I just they put their hand down. N. Anthony Raj, I did see your hand go up.
Please go ahead and unmute, ask your question.
Yes, Anthony Raj, I see your hand. You need to unmute and then you can ask your question, or you can type it.
Please unmute. Yeah.
I don't know.
Anthony, can you unmute your button and then ask the question, please? Maybe there is a technical problem or something.
Yeah. And students, please ask questions, kids. Don't worry about, in India, we always worry about, oh, am I asking the right question? Am I drawing attention to myself? Don't worry about that. No question is wrong. Every question is worth an answer. So ask whatever you want, you should ask.
தமிழ்ல
You know,
கேட்கனாலும் கேளுங்க.
correct. Yeah, that is I want to say. If anyone want to ask, he can ask in Tamil as well.
பட் நான் தமிழ்ல ரிப்ளை பண்ணுவேனான்னு எனக்கு சரியா தெரியல, பட் ட்ரை பண்றேன்.
That's perfectly all right. Yeah. Well, what happened to Anthony Raj? Can you unmute? I think they're having some technical problems and all that.
Yeah.
Let me see. The time is now. To tell you what, if you don't mind, send your questions on email, and I will forward it to Dr. Lakshminarayan so that he can answer those questions in case you have some. Swatish, can you please unmute? Ask your question.
Yes, sir.
Go ahead, Swatish. Can't hear you at all. Can you unmute, Swatish? Lot of background noise, Swatish. Sorry, there's a lot of disturbance. I'm sorry, I can't hear you at all. Swati?
You can type, if you like, you can type the question in chat, then we can read your question.
There are lots of network issues in different places. My apologies. Yeah, there seems to be some problem with Swatish unable to. Swatish, can you please cut your mute mic off? There's a lot of disturbance. But anyway, so please send your questions by email, and we will make sure that Dr. Lakshminarayan is able to answer. You know, there were hundreds and hundreds of questions sent to Dr. Sivadan Pillai. I am so thankful to Dr. Pillai for answering every one of them. Same way, you will get all the answers back if you can send us your questions either by chat or by email. The email is msvijay2020@weservefoundation.org. All right, I can, you know, or you can, Dr. Kachuru, would you mind giving your email in the chat? That way they can send it to you as well. You can put your email in the chat that children can access it. And I think it's getting a little bit close to time.
Yeah, my email is in the chat, so feel free to send me questions also.
Okay.
You can even write to me. That's not a problem. I'll send you my email. Feel free to write to me.
In that way, right to Dr. Lakshminarayan? I'm sorry, we have some technical issues at various places. Look, now again, based on where you are, sometimes the network is not connecting very well. I am honored that Dr. Pillai could join us today. And again, this he explored the entire universe for us. He brought the universe to us. Today
This thing we learn a lot. That's
Yeah.
why
we're looking at from the microscopic point of view, how so much. Dr. Lakshminarayan, again, it's a fabulous knowledge you have shared with us, sir. Hope that has motivated a lot of youngsters. And I wish I were a young fellow now. Anyway, well, again, Santosh, can you unmute? Santosh, unmute and ask your question, please.
Yeah, there are two hands up, I see.
Yeah. I don't know why they're not able to unmute and then ask the question today. I'm asking you to unmute, please.
Also, Sanjeev has his hand up.
Sanjeev, can you unmute and ask your question?
I
Sometimes
think sometimes accidentally.
Sometimes the settings also, they don't know how to go back in the settings. Then the privacy, they can microphone, they need to unmute. I also face the same problem. Maybe first time they trying and then they don't know how to make it sudden.
Well, again, if you don't mind, if you're not able to unmute and ask the question, please go ahead and send your question by chat, and Dr. Kachuru might be able to read it to us. And are there any questions in the chat box so far?
I don't see anything now.
Well, okay. Well, again, I think it's getting late for the children also to go back to their routine, whatever they need to do. Dr. Kachuru, would you kindly help us conclude the session with a...
So thank you everyone for your time and attention and, helping us run this program. And of course, Lakshmi, I can't thank you enough. I myself learned so much. I had forgotten a lot of things. So thank you for reminding me of all of that. And thank you for reminding us what a fascinating world there is, beyond our eye, what our eye cannot see. You covered, like I said, just a tremendous breadth of topics, and for the students, that just tells you, if you want to emulate somebody, be like Dr. Iyer. Ever since I have known him, he has been curious. Anything he's curious about, he makes sure he makes the effort to find out or read about it, and that has inspired me also personally. So thank you again for a wonderful talk, and thank you, Dr. Vijay, for arranging this, and all the other eminent scientists, and it's my honor to meet everybody. Thank you.
Good evening. Have a wonderful evening, all of you friends. And once again, thank you everyone for participating.
Thank you.
Thank you, Dr.
Thank you so
Iyer.
much.
Thank you, sir.
Thank you so much.
Thank you, sir.
Thank you so much.
Thank you, sir.
I think
Thank you,
Dr.
sir.
Vijay wants to
Thank you,
say
sir.
something.
Thank you, sir.
Thank you. Thank you. Very nice.
Nandri, sir.
Nandri.
Very good. Nice to hear from all of you. Very nice. Well, I will conclude today. Thanks again.
Okay.
for participating.